Okay, so yes, we are going to be covering stem cell research and cloning, but even though it is perceived to be a very technically heavy subject, chances are you won't find that's the case. I'm going to try to simplify it in a way that makes sense and it's practical for us. But before I begin, we have to sort of settle some groundwork, okay? And that is, I want to ask you, why is it that you believe abortion's wrong? What is the pro-life view with regards to abortion? Can somebody give me just a rough explanation? Yes? Okay, perfect. So she said basically she thinks abortion is tantamount to murder. It's killing an innocent human being. And as you said, you believe that the unborn is the same inside the womb as it is outside the womb, okay, basically. So, and that's exactly right. So the reason why we're against abortion is because abortion kills an innocent human being. Therefore, we think that the unborn is a human being, an innocent human being, just like you and me. Now, you probably have reasons why you think that's true. What scientific arguments you might have for thinking that the unborn is a human being. Now, we're not going to go over those because we're going to be talking about stem cell research and cloning, but I want you to think with yourself, if you will, for a moment, and just think, okay, I have reasons why I think it's wrong to kill the unborn because the unborn is a human being. Well, we're not going to be talking about the unborn in the same way because obviously with stem cell research and cloning, we're not talking about something that's in the womb, so it's not unborn yet. But we are still talking about a developing human being from the very first moments of its conception. So in other words, whatever arguments and evidence you have against abortion will be able to be carried over onto this particular topic. All right? All you got to do is be able to understand what is stem cell research, what kinds of things are going on, and then you'll be good to go. And so that's what I propose to do with this talk, is to give you a basic understanding of what's going on in the world of stem cell research and cloning. Now, the body is composed of about 200 different types of specialized cells, okay? And when I say specialized, I just mean that these cells have been given a specific task, a specific function. So we got bone cells, brain cells, blood cells, pituitary cells, cartilage cells, heart cells, lung cells. Notice every one of these types of cells has already been given a specific function of what it's supposed to do, okay? That's what these specialized cells are, and you have over 200 different types of specialized cells. A stem cell is simply an unspecialized type of cell. In other words, it has not yet been given a specific task. It hasn't been told, okay, I want you to be a stomach cell, or I want you to be a heart cell, or I want you to be a brain cell. That's all a stem cell is. It's kind of like a general cell that has not been assigned as to what type of cell it's going to be in your body. Now, these unspecialized cells become specialized cells through a process known as differentiation. Okay? So a stem cell, which again is a general cell with no specific function, becomes a specific type of cell through the process of differentiation. And so if you had a stem cell here, and here's just an example, through the process of differentiation, it becomes a specialized cell. And you'll notice in this case, I've just, as an example, shown it turning into a heart cell. Okay? So again, this is a generalized, unspecialized cell. This is a stem cell. And it becomes a heart cell through this process of differentiation. Notice also that when it differentiates, it also creates another one of itself. Okay? So stem cells are said to be self-renewing. They might become specialized, but in that process, they also develop another one of themselves, which is quite handy. Now, what kind of stem cells are there? Well, there's about 4 different kinds. There's unipotent stem cells, and these stem cells can turn into one type of specialized cell. So maybe this kind of a stem cell can only turn into a bone cell, and that's it. All right? There's also multipotent stem cells, and these stem cells have the potential to turn into a few closely related types of cells, like maybe blood cells. Notice, multipotent stem cells are kind of more powerful, if you will, than a unipotent stem cell because they can turn into a number of different types of cells. Then you also have pluripotent stem cells, and these stem cells can turn into any cell type that's found in the human body. So any one of those over 200 different types of cells. That's pretty powerful, okay? And then there's totipotent stem cells, and these can turn into any cell type that's found in the body or into any cell type that's found in the supporting tissues of embryonic development. So for example, the placenta or the umbilical cord. Now, of these 4 types of cells, which do you think scientists are most concerned about and most interested in using for research? What do you think? Yeah, I heard totipotent. So yeah, you think so. You think that they can, wow, we want totipotent stem cells because they have the most power. But in reality, they're more concerned about pluripotent stem cells. It's these stem cells that they're interested in extracting and using for certain research. Now, where do scientists get these pluripotent stem cells? Well, they get them from a human being within the first week of development. In fact, this is a selfie I took of myself when I was just 6 days old. You can't see the camera because I was using a selfie stick. But no, this is what we all look like at about 6 or so days old, and then we call this a blastocyst. So blastocyst is simply a human being within the first week of development. All right. Now, this is obviously a computer-generated drawing, but this is what it actually looks like for real. If you had a scanning electron microscope or a really powerful microscope, you'd see this is what a human being looks like at an early stage of development. Now, if you were to cut that blastocyst in half and look at the inside, you'll notice this picture here, which shows in the side of it a whole conglomeration of cells right there, and it's labeled ICM. That stands for inner cell mass. Those cells conglomerated on the edge there are pluripotent stem cells. So this is where scientists get pluripotent stem cells, from a developing human being at the blastocyst stage. They're all just conglomerated right there. Now, what do scientists want to do with these pluripotent stem cells? Well, pretty simple. They basically want to take the developing human embryo, again at the blastocyst stage, extract those pluripotent stem cells, then coax those stem cells to turn into some specific type of tissue. So in this example, I've shown these stem cells being coaxed into turning into heart muscle tissue. And then once they have this new tissue, they want to implant it into a person's, well, in this case, a heart. So pretend, for example, this person had a heart, well, has a heart, and they had a heart attack. And part of this portion of their heart died. That's what happens in a heart attack. Part of the muscle tissue dies, which of course causes it to not function as efficiently. Obviously, if your heart attack is large enough and you have a lot of that tissue die, then of course you will die because your heart won't work at all. But if you have a minor heart attack, it might just cause some portion of your heart to die. And so therefore, if you had a developing human embryo and had their stem cells taken out and had some of these stem cells turned into heart muscle tissue, then you could have it implanted and repair your damaged heart. Wouldn't that be great? Okay. So just for sake of illustration, I need kind of a volunteer to just so I can pick on that might have a damage. Yes, thank you, sir. What's your name, by the way? Logan. You don't have to go. You don't have to come up. I'm not going to hurt your heart. You can sit down, but I just need to point to you. Okay. So we're just going to pretend Logan has had a small heart attack. Okay, and part of his heart muscle tissue has died. Thank you. He's obviously a theater arts major. Give that man an Oscar. So suppose he had heart muscle tissue die, and then we were to take a developing human embryo, take its stem cells, coax them into turning to heart muscle tissue, and then we implant it into Logan's heart where his damaged tissue is. Okay, that would help his heart function better. But what problem might arise if we took this tissue and implanted it in his heart?
Body rejection.
Body rejection, that's right. His immune system would say, wait a minute, you're not Logan's body, you're not Logan's tissue, and thank you for making my heart work better, but we're going to send an immune response and we're going to attack that new tissue that's implanted on his heart. Okay? And that's the problem with embryonic stem cell research, is that when you implant it in a person's body, your body's immune system will reject it because it recognizes it's a foreign body, it's a foreign invader, if you will. So how do we solve this problem? Well, this is where cloning comes into place. Okay? So embryonic stem cell research creates the problem of tissue rejection. But if we could somehow create an embryo that was identically matched to Logan's DNA, well then, of course, when we create new muscle tissue and implant it in Logan's heart, well then his body would not reject it because it would perceive that tissue to simply be part of Logan's body. And so this is why cloning is so needed in the embryonic stem cell research realm, because it solves the problem of tissue rejection that's created through embryonic stem cell research. Now, you might say, well, what exactly is cloning? Well, cloning is simply a form of reproduction. Okay? There's many different ways to create a human being. For example, you've probably heard of in vitro fertilization. This is where you take a sperm and an egg, and in like a petri dish, you put the sperm and an egg together, and that can create a new human being. You can also create a human being through sexual intercourse. Rumor has it that's still popular, okay? And people still do it today. That's another way to create a human being. But a third way to create a human being is through cloning. Again, it's simply a form of reproduction. It's just another way to create a human being. And the most common method that's used today for cloning is called somatic cell nuclear transfer. And that's a very fancy term for a 3-step process that I'm about to show you in a minute here. Don't try this at home, okay? But here's what we got to do. Oh, we need to volunteer, a female preferably. Yes, thank you. What's your name? Caitlin. Okay, so Caitlin, out of the generosity of her heart, you must be Christian because you're going to help Logan out. Are you? Fantastic. Thank you so much. All right, so Caitlin, it's Caitlin, right? Yeah, so we need one of Caitlin's eggs to get this whole process started. So we're going to extract an egg from Caitlin. Not right now, don't worry. Just hang on tight. It'll hurt later, but no problem. So we're going to take one of her eggs, but now Caitlin has her own DNA found in that egg. Okay. Now, how many chromosomes of Caitlin are in her eggs? 23 chromosomes, that's right. How many chromosomes are in all of our cells, normally? 46, that's right. So you get 23 chromosomes from the egg, 23 chromosomes from the sperm. They come together, you get 46 chromosomes. Except for our sperm and egg cells. Those, of course, only have 23. So we have 23 chromosomes in Caitlin's DNA, but we don't want that because we want a genetic duplicate of Logan. So the first step in somatic cell nuclear transfer is to remove your DNA. Bye-bye, it's gone. Okay, no offense taken, okay? So then we want to take some of Logan's DNA and insert it in Caitlin's egg. Now, where would we get his DNA from? Well, we can get it from any one of his body cells because all of his body cells have 46 chromosomes. So we're just going to take a little cell from your skin, if that's okay. You don't mind? Okay. Thank you very much. Very generous as well. You guys are both generous. You guys are generous people in this crowd. So we're going to take your cell from your arm, extract the DNA from it, and then we're going to implant it in Caitlin's egg that's had its DNA removed. So there's Logan's DNA going into Caitlin's eggs, all right? Egg. So now we have all 46 chromosomes of Logan's DNA in the egg from Caitlin that's been denucleated. The nucleus has been taken out. And then all we're going to do is give it a little bit of electric shock, and now that egg that has 46 chromosomes from Logan will start to reproduce and grow, just like a human being who has been fertilized through sperm and egg would also start to grow. So now we've created a genetic duplicate of Logan. In fact, let me show you a video of this taking place for real. So this is an egg, and this is a little vacuum pump that's going to hold the egg next to it. So there you can see the vacuum turn on. And this functions like a drill and like a pump and a tube and a suction and a whole bunch of things. Okay, so here's what it's going to do. So right there, just cut a hole in the outside of the egg. And it's going to take that material from that hole and spit it out. Now it's going to jump in there and it's going to suck out the DNA. So there goes Caitlin's DNA. It's gone. Right? And it's going to spit out her DNA on the outside of the egg. There it goes. So there's Caitlin's DNA stuck on the outside. So now this is a denucleated egg. And of course, they'll do a whole bunch of these in the process. Now we want to take Logan's DNA, which he's conveniently donated, of course, for his own benefit, so it's not really that much of a great thing. But here's what it's going to do. It's going to cut a hole in the outside of the egg. There it spits out the hole. And now you can see right there, there's Logan's DNA coming right in. And it's going to inject it right into the egg. And it just did. Puts it right there. And now they have just cloned a human being. So right there, you saw a human being created through cloning. And it just does the same thing again. Moves it over, suction machine turns on, spits out a little part of the outside of the egg, and there's more DNA being inserted into another egg. There was another human being just cloned for real. Okay? So as you can see, it's, in a sense, kind of a simple process. All right? They're just doing it like a machine. All right? So that's what cloning is. It's creating a genetic duplicate of another human being, and specifically in this case, we're doing it with Logan. Now, there's two different types of cloning. The first is reproductive cloning. So say, for example, we really like Logan. He's a cool guy, he's funny, he's intelligent, he's charming, he's got a great haircut today. We have a lot to give him credit for. So we want a lot of Logans. So we could do that process and then take that clone that was just created through somatic cell nuclear transfer and implant it in a woman's uterus. Okay? So we'll put it in Carly's. Appreciate it, Carly, because Carly was at Camp Joshua about a month ago, and she was very generous as well there. So she's going to lease out her uterus. And so we're going to take that clone, put it in Carly's uterus, and grow it till it's full term, and it'll be born. And then we'll have another little Logan running around. So notice, with reproductive cloning, it creates not just a genetic duplicate of another human being, but if we allow that genetic duplicate to grow to term, it's like creating another person that's like Logan. Would that clone have a soul? Does anyone have an answer? Yeah. You say yes. Okay. Is anyone here an identical twin?
They run in my family.
They run in your family. So do you know any identical twins?
I know identical twins and identical triplets.
You do know identical twins. This is very interesting. Now, you've met both of them, right?
Yes.
Do both of them have a soul?
Yes.
You sure? Yes. Okay. So both identical twins, per her report, and I've met some identical twins as well, so I know this to be true, they both have souls. Okay? So even though you can have a genetic duplicate of another person present in the same room, both of them will have souls. And indeed, in this case, if you cloned Logan, his clone would also have its own soul. Because it's just like creating twins, or triplets, what you said in your case. Okay? Except in this case, this twin will be, how old are you, Logan? 19. So he'll have an identical twin that's 19 years younger than him, which would be kind of creepy, wouldn't it? But nevertheless, or interesting, depending on your perspective. All right? So that's what reproductive cloning is. Now, there's another type of cloning. And by the way, reproductive cloning is, as you can imagine, fairly unwelcome at this point in our history of life, although I'm sure in time this will become a little bit more interesting and intriguing to a lot of people. But we also have what's called therapeutic cloning. And therapeutic cloning involves exactly the same process of cloning. In other words, it's also somatic cell nuclear transfer. You're also creating another genetic duplicate of a human being like Logan, although in this case, instead of implanting that human into Carly's uterus for it to grow to term, we are going to kill that developing human being at the blastocyst stage, take its pluripotent stem cells out, and use those stem cells to coax them into some tissue that we can later implant into Logan's body. And that way, that new tissue will not be rejected by Logan's immune system. Okay? That's what therapeutic cloning is. So in other words, therapeutic cloning and reproductive cloning entail exactly the same thing. The only difference is that with therapeutic cloning, we kill the developing human being before it ever grows to term. That's the only difference. Otherwise, it's exactly the same technical process. Yes?
And does that one have a soul?
Does that one have a soul, the one that we kill before it reaches term? I would argue yes. I would argue that as soon as you have conception, as soon as you have 46 chromosomes, you have now a soul. Now, if you're of a person with religious convictions, there are two different types of views about when ensoulment occurs. So there's a view called creationism, not to be confused with like creation of the universe. But creationism is the theological position that God creates a soul ex nihilo, out of nothing, at either the moment of conception or at some point before birth. Okay? So if you hold to a creationist viewpoint of ensoulment, then it's possible that you could argue there's no soul at conception. But then later it comes out. God creates a soul and puts it in. There's another view called traducianism. And this is the view that the soul arises at the moment of conception as a result of the union of the man and the woman. And if you hold to the traducianist view, you would hold that ensoulment occurs at conception and no later. It has to occur at that point. Now there's traducianism, Traducianism. Traducianism. So it's creationism, traducianism. So there's pros and cons to each theological position, but I'm not going to get into it right now, but those are the two different views. Yes?
So with reproductive cloning,
The scientific medical benefits. True, but the thing is that even with reproductive cloning, if somebody's lost a child and they would like to have another one of the same, perhaps they would, or maybe you want to clone yourself because you're just really into yourself. I don't know. Yes? Has reproductive cloning ever been completed? Well, it has with animals. You've probably heard of Dolly the sheep. Dolly the sheep was the first animal that was cloned, but for some reason we haven't been able to perfect it with humans just yet. I'm sure it will happen in time, but at this point we don't have any clones that we know of that are walking around and talking just yet. For some reason it's a little bit more complicated and they tend to die much sooner.
I think they're working on that up at OHSU now.
Oh yeah, I mean there's lots of countries around the world that are working on it, absolutely.
and there was a problem with Dolly. What was the problem with Dolly?
The problem with Dolly was that she died of degenerative diseases at a much earlier age than one would expect. So, for example, if that was to draw a parallel with humans, oftentimes we see things like heart attacks or coronary artery disease or stroke or arthritis in humans that are, say, 40, 50, 60, 70 years old. But what we saw with Dolly was these kinds of degenerative diseases at a much younger age, say like 20 years old, 30 years old in human years. I don't know what it is in sheep years, but you get the point. So there's something going on with why Dolly's died at a much earlier age than normal and developed these kinds of diseases. So just to summarize what we're talking about, cloning creates the embryos that are matched to Logan's body or whoever the patient is. Embryonic stem cell research takes the clone, takes its stem cells, and uses it for scientific or medical benefits to benefit Logan. All right? So that's why the two sort of need each other in order to work out well. Now, what are some of the benefits of this type of research, embryonic stem cell research and cloning? Well, here's one benefit. They have the potential to treat disease and disability. So, for example, heart attack, stroke, Parkinson's, paraplegia, arthritis, you name it. Is this not a worthwhile goal to help cure and address these ailments? Is this not a noble goal? Yeah, sure, it is. I mean, just strictly speaking, that's certainly a worthwhile thing. We'll get to the questions of whether we ought to do that or not for reasons in a minute. A lot of people say also we have a moral requirement, obligation to perform this research. This is Senator Tom Harkin of Iowa. Actually, he just retired, I think, last year. But he even says it's morally wrong to oppose funding for this kind of research because the embryos we're talking about are no bigger than a period at the end of a sentence. And so therefore, they don't have the capacity to become a human being. Is that true? No, it's not true. Well, is it true that they're no bigger than the period at the end of a sentence? Yeah, it's true. But what do you think about the statement, they don't have the capacity to become a human being? Do they have the capacity to become a human being?
They already are.
That's right, they already are.
Maybe not.
So the answer would be actually no, they don't have the capacity to become a human being because they already are human beings just at a very early stage of development. Okay? So again, now we're going back to our pro-life apologetics, pro-life defense views. What reasons do we have to oppose abortion even from conception? Well, because they're a human being already. And so this is why that same argumentation can be applied even to embryonic stem cell research and cloning. Some Christians argue that we have a theological mandate to engage in this training. In fact, these two guys who published an article in The Scientist said, look, salvation includes healing of the body and the soul. And we emulate God when we heal through agape love. Right? And so therefore, this research, stem cell research and cloning, basically contributes to God's healing on earth. After all, didn't Jesus heal people's bodies when he performed his ministry on earth? So aren't we doing the same thing?
So
I see if
embryonic stem cell.
No, he didn't use embryonic stem cells, but we have to do that. So wouldn't that, in essence, be the same thing as what Jesus did?
No, he didn't trade a life for another life.
Ah, okay, that's right. Yeah, Jesus didn't kill another human being, innocent human being, for the sake of healing somebody else, right? He was just able to supernaturally do it. Okay, so you're right. But nevertheless, you can see even Christians make this argument that we need to be engaging this type of research. Michael Kinsley, a well-known columnist, says, look, the embryos used in embryonic stem cell research and cloning are not human beings. And here's his argument. He says, look, we're talking here about newly formed embryos. These are not fetuses with tiny waving hands and feet. These are microscopic groupings of a few differentiated cells, and there is nothing human about them. So let me ask you this. Is it true that we're talking about newly formed embryos? Correct. He's right there. These are not fetuses with tiny waving hands and feet. Is that true as well? Yeah. The reason is because we kill them before they ever develop hands and feet. But that point aside here. These are microscopic groupings of a few differentiated cells. Is that true? Yeah, that's also true. There's nothing human about them. Is that true? No, it's false. It's everything human about. They are human beings, okay? So, but that's strange. Wait a minute. Michael Kinsley posts articles in Time and Newsweek and national magazines. Are you saying he's getting his science facts wrong? Yes, he is. Okay, so obviously it goes to say not everything you read in Time, Newsweek, or Wikipedia for that matter, is true, right? Mary Tyler Moore, who's an actress, for those of you who are my age or older probably know who she is, and those of you probably younger may not know who she is. But nevertheless, she's an actress, and she appeared before Congress because she has what's called type 1 diabetes, which is juvenile diabetes. And she was arguing in favor of this kind of research, embryonic stem cell research and cloning. And here's what she said. The embryos that we're talking about bear as much resemblance to a human being as a goldfish. Okay. So that's the extent of her argument there. Now, of course, it doesn't really matter what it looks like, right? I mean, obviously, at the blastocyst stage, we don't look like you and I do, but that's because human beings aren't supposed to look like you and me and have arms and legs and hair and body odor when we're just 7 days old, okay? So human beings, in fact, all living things look different at different stages of development, all right? And so when somebody says to me, well, Alan, the blastocyst doesn't look like a human being, my answer is, that's false. It looks exactly like a human being. It looks exactly the way a human being is supposed to look at that stage of development, right? So, if you saw a monarch and it was just a little egg, you'd say, that's not a monarch, but it is. And then when it's a caterpillar, it's still a monarch. And then when it becomes a chrysalis, it's still a monarch. And then when it becomes a butterfly, it's still a monarch. Because living things look different at different stages of development, yet they're still the same thing all the way through. Ron Reagan, the son of President Ronald Reagan, spoke at the 2004 Democratic National Convention, which was a surprise to a lot of people. But he also got up there and on national television argued that we should be in favor of this research. He said, these cells could theoretically have the potential, under very different circumstances, to develop into human beings. That potential is where the magic lies. But they are not, in and of themselves, human beings. They have no fingers, no toes, no brain or spinal cord. They have no thoughts, no fears. They feel no pain. So what do you think of Ron Reagan's comments? Is he correct? Yeah, there's a few sentences. He's correct. They don't have any thoughts or fears. They don't feel pain. That's true. That's because we kill them before they ever develop those things. But yet he said on national television, they are not in and of themselves human beings. How could he say something on national TV that's just scientifically wrong? Is that possible? Yes, it's possible. It happens all the time, okay? And unfortunately, he made this comment as well. Read the whole, you can read his whole case, but here he talks about here's how this process works. He says, imagine going to a doctor who, instead of prescribing drugs, takes a few skin cells from your arm, like we did with Logan, and the nucleus of one of your cells is placed into a donor egg whose own nucleus has been removed. Sounds like what we did with Caitlin, right? A bit of chemical or electrical stimulation will encourage your cell's nucleus to begin dividing, creating new cells, which will then be placed into a culture. What did Ron Reagan just now describe? What's that? Cloning, that's right. He just described somatic cell nuclear transfer. But if you read the transcript of his speech, you know what one word you won't read anywhere in there? Cloning. Why? Because that would freak people out. If he says, we should pass this bill, which was, $22 million for cloning. If people said that, they said, no, we're not going to vote for that. Because he knows that's not going to sell. So he just describes it as a medical procedure, scientific procedure that will help you with certain things. He goes on to say, look, these cells will generate stem cells. How would you like to have your own personal biological repair kit standing by at the hospital? Sound like magic? Welcome to the future of medicine. So he envisions this clone of Logan sitting somewhere waiting for him so that when he gets sick, he can go to it, take its body parts, and use it for his own medical benefit.
That was the movie The Island.
Yep, hold your thought. We're going to get to it. Thank you. So he goes on to say a bunch of this other stuff, all right? Now, the case for embryonic stem cell research and cloning is flawed for many reasons, which we don't have time to go into all of them, but I'll just point you to the most obvious one. And that is, you have to kill an innocent human being in order to harvest their stem cells. Why? Because a human being uses their pluripotent stem cells to create its body. And if you take those pluripotent stem cells from the developing human being, it cannot create its body. Therefore, it dies. And so this way, if you know your arguments, your scientific defense against abortion, then that same argument and evidence can be applied to the human being at the early stage of development, even though it's not in the womb at the point. Okay? So that's the main reason. There's other reasons why we, of course, read against it, but that's the main reason, at least the most important one.
Can I ask one question?
Yeah, so fertilize is kind of the wrong term to use because we're not actually taking sperm and fertilizing the egg with it. We're taking 46 chromosomes from Logan and implanting them in an egg that no longer has its DNA. The net result is the same as if you put a sperm and an egg together.
So that egg acts as a host.
The egg acts as a host. Yeah, so when you get sperm and egg together through normal sexual reproduction, the sperm gives 23 chromosomes to the egg, and they get 46 chromosomes. So you have one cell now, a zygote, which is a human being at the first stage, with 46 chromosomes. Somatic cell nuclear transfer or cloning does the exact same thing, except it achieves it in a different method. It just takes an egg with 23 chromosomes, removes it, takes 46 chromosomes from one of Logan's body cells and puts it right in there. And that way you end up, again, with a cell with 46 chromosomes, a zygote.
All from the
So it
same
kind of
person.
skips the stage.
It skips the sexual reproduction stage, it skips the sperm coming to the egg. And instead of doing 23 and 23, we just take...
That's what creates the sorry.
No, in the process, no, that's okay. In the process of creating that clone, you're right, they don't kill anything. The problem is once they've created the clone, and now we have an identical twin of Logan, then to take the stem cells from that, they have to kill that newly developed human being in order to take its stem cells, and then we take those stem cells and create new tissue for Logan's body.
Okay, thank you.
So cloning just creates the human being. Embryonic stem cell research then kills the human being and takes it. It's okay. It's a legitimate point of confusion there. Now, some people say, well, Alan, look, we already have through fertility clinics today, because lots of people go through in vitro fertilization, parents who can't have kids, they produce all these embryos so they can implant them in the wife, but once they're done with that process, they have all these leftover human beings that are now sitting in freezers all across the United States. There's probably 400,000, if not more by now. This data was taken a few years back. But we probably have 1 million human beings frozen at about 5 days old, sitting in freezers all over the country, perhaps all over the world. And so they're going to die anyways. Scientists are saying, let us take those frozen embryos, take out their stem cells, and use them for medical research. Now, what's the problem with doing that? Yeah, you're killing all those frozen human beings, okay? So another alternative to dealing with all these frozen human embryos, this would be my suggestion and many other people's suggestion, is to adopt them out. According to the Centers for Disease Control, there's 2.1 million infertile couples and 9.3 million women who are using infertility services, meaning they're trying to get pregnant and they can't. So my suggestion is, why don't we allow this huge demand of people who are longing to get pregnant have access to these frozen embryos, adopt them out, basically adopt them, thaw them out, place them in their uterus, and have them have a chance at life. In fact, this has actually been going on for 20 years about. And I know a couple of my friends, very close friends, who actually did this very thing. This is Catherine and Claire. I'm not sure actually which one's which, but let's just pretend this is Catherine and this is Claire. These are two people that I know today. Okay? They were frozen at 3 days old. Let me see here. Catherine was frozen, at 3 days old for 5 years, and her sister Claire was frozen for 8 years. And so my friends adopted these two frozen embryos that were sitting in a frozen fertility clinic, had them thawed out, placed them in my friend's, the woman's, uterus, and eventually they grew to term, they became born, and now they are two beautiful little girls that run around and play with my kids, and are just as normal like any other kid. Now, when you touch them, they feel a little bit cool to the touch, but apart from that, they're just normal, right? And so my suggestion is, why don't we give these kids a chance at life rather than killing them and using their body parts for spare parts for other human beings? Why don't we give them a chance at life? This works perfectly well. In fact, it's rather successful, okay? Now, some might say, well, Alan, so don't you think cloning, though, is kind of weird and creepy? And I think the answer is yes, I do. But when speaking to secular audiences, you can't just say, well, this is weird and creepy and you're playing God. And so my concern with cloning isn't so much how a person comes into existence, meaning whether it's through in vitro or sexual reproduction or cloning. My concern is with what we treat or how we treat the clone once they come into existence. Because every case in which a clone is created, they will always be treated as less than valuable, less than human. And that's my concern. In fact, as you kind of mentioned already, there's a movie that depicts this quite well. How many have ever seen this movie, The Island? Raise your hand. Oh, man. So if you haven't seen The Island, and your parents let you, you should see this. Because this movie actually raises this exact issue that I'm talking about, cloning and harvesting people's body parts for other people's health, in a way that's very persuasive, and it actually takes our side on the issue. It actually argues that this is actually immoral. In fact, let me show you a video clip from the movie. And in this particular scene, two of the clones, who are like adults, they're walking around, talking, have escaped from this underground facility. They think that they're sitting in this underground facility because the earth has been diseased with some bacteria. And so they think they're the only survivors. But they don't know that actually the world's going on about them, and people who are rich and famous have actually created clones of themselves and are being raised in this underground facility. And once these people who are out in the real world get sick, they're going to take the body parts from their clones and put them in them. But these clones don't know that. Nevertheless, they happen to escape and run to a nearby town where they meet one of the employees of that facility. And this employee now describes who they are. Take a look and see what they say.
Why do they lie? Why do they lie to us? Tell me. To keep you from knowing what you are. What we are.
What are we?
Oh, man, why do I gotta be the guy who tells the kids there's no Santa Claus? Okay, look, you're, well, you're not like me. I mean, you're not, human. I mean, you're human, but you're not real. You're not like a real person, like me. You're clones. You're copies of people out here in the world.
What?
Clones?
What?
Copies? What are you talking about? Some hag trophy wife needs new skin for a facelift, or one of them gets sick and they need a new heart. They take it from you. The whole reason you exist is because everyone wants to live forever. It's the new American dream. And there's people out there that are rich enough to pay anything for it.
Don't people care that they kill us? That they take parts from us?
They don't know. They think you're vegetables simmering in a jelly sack. Why do you think Merrick has you stuck in an old military bunker below ground? He doesn't want anybody knowing the truth, especially not your sponsors.
Sponsors?
The people that had you made. They, own you.
But why doesn't Merrick want our owners to know that we're alive?
Just because people want to eat the burger doesn't mean they want to meet the cow.
So very interesting that this movie, again, takes up this question and actually takes our side on it. They show how crazy and inhumane it is to create these human beings and then strip them of their body parts, killing them, of course, and use them for other people's benefits. Okay, amazing. In fact, it's interesting they even say the people who had these clones made think that you're just vegetables simmering in a jelly sack. And in fact, this is exactly the way abortion choice advocates think of the unborn. It's just cells, unspecified cells. They don't realize it's a human being, and the same thing is going on with this. So again, if you have the chance, check out this movie. So now the question is then, is this the only way we can treat disease and disability? Do we have to kill human beings in order to do that? And the answer is no. There's actually many different ways we can treat disease and disability without killing innocent human beings. And the most powerful way, using a similar kind of power in research, is with adult stem cells. And adult stem cells are Exactly what you think. Stem cells found not from embryos, but in adult human beings like you and me. In fact, we have stem cells all over our body, in our skin, in our eyeballs, in our bone, in our fat, everywhere. Okay? And there's a tremendous number of benefits using these particular stem cells. Number one, innocent human beings don't have to be killed. Woohoo! We can take stem cells from Logan's body without killing him. We just give him a little pinch on the arm, yank out some stem cells that are sitting there, because after all, when Logan cuts his skin, what happens to his skin? It grows back. Why? Because remember, stem cells are self-renewing. They just reproduce themselves, so they create new skin for Logan. And so we can take them and coax them into turning into whatever tissues we want. Also, consent can be granted, right? You can't ask a blastocyst, excuse me, do you mind if we take your stem cells? But we can ask Logan, excuse me, is it okay if we take your stem cells? It'll save your life. Look at that, he said absolutely. Okay, so we can get consent. We also don't have to clone human beings. No need to create a clone of Logan, because after all, if we take stem cells from his own body, coax them into turning into new tissue, like heart muscle tissue, implanting it back into Logan's body, then his body would say, that's just my own body. What's the big deal? And it won't reject his new tissue because it's from his own body. So you can skip the whole cloning process completely. Also, we eliminate the potential harm to women who donate eggs. Look, getting eggs from women is not as easy as getting sperm from men. It requires a surgical procedure. And in fact, there's a whole documentary called Eggsploitation, which shows how many women will be exploited and are exploited for their eggs because, of course, this is a very valuable commodity. Also, and I'll close with this, is that there are a lot more promise to adult stem cells that have shown already themselves to be very powerful in treating diseases. I'll just give you a couple of examples. This is a study done in the New England Journal of Medicine where they took people who had blindness because the cornea, the outer layer of their eye, had become opaque. So now they're blind in that eye. So here's what they did. They took stem cells from the other person's eye, from the other eye in their head, and they grew a new cornea on an amniotic membrane. They took that cornea, took it out of the amniotic membrane, cut out the old cornea, took the new cornea, put it on there, and they fixed blindness. This is back in 2000, 16 years ago, they were treating blindness with the people's own adult stem cells. Now they've perfected this procedure. This is a study that was done in Italy where they took, instead of now growing the cornea on a separate amniotic membrane, they simply take the person's cornea, cut it right off, and then literally implant new adult stem cells right onto their eyeball. And it grows a new cornea automatically. Treatment worked completely in 82 out of 107 eyes. It worked partially for 14 others, and a person with severely damaged eyesight for 60 years has now near normal vision through adult stem cells. Awesome. Bartolo Colon, if you're a New York Yankees fan, well, I don't think he plays for the Yankees anymore, but as you know, a lot of pitchers, after a lot of times pitching like this, they tear the ligaments in their elbow and their shoulder. No problem. They took some adult stem cells, pulled them out of his fat and his bone marrow cells, implanted them directly into his ligaments, and it grew new ligaments, and he was able to go back to pitching afterwards. Awesome. It's also used for heart attack stem cells. So what they used to do is if a portion of your heart died, this is the bottom portion because there's a blockage in the artery, so blood cannot get to this bottom portion of the heart, so it dies. So here's what they do. They take stem cells from the person's hip bone, they clean them out, they put them in a syringe, and then they inject them into the artery. The stem cells go down here, they start to perfuse in this damaged tissue of the heart, and it regrows heart muscle tissue. And it fixes their heart attack. Look, they were doing this 14 years ago using adult stem cells. Now they've perfected the procedure. They take stem cells directly from the person's bone, and instead of it running it through the artery, they inject the stem cells directly into the person's damaged heart, and it regrows new muscle tissue. Fantastic. One last example. This is a guy who had a cancerous tumor growing in his trachea. This is his windpipe. It was going to grow so much it was going to block off his windpipe so he couldn't breathe. He would suffocate and die. People said, all right, this guy's toast, no chance. Some people said, no, we have an idea. They took his stem cells, they created a scaffolding in the shape of his trachea, they grew his stem cells to create a new trachea, they took out his old trachea, put it in a new trachea, and now he's got a normal life expectancy. This is science fiction that's been going on for decades using adult stem cell research. And in fact, adult stem cell research has been used in human beings with success in over 73 different types of applications. Embryonic stem cells, well, it used to be 0, but now they actually have 3 applications that have, with minimal results, worked in humans. But for the most part, adult stem cells have been far more successful in treating human beings.